out on Herc?
Yep. Ready for control. Bumping. Bumped. Roger. Roger, thank you, Captain.
Wait till you catch up.
Thank you.
You can shoot for 25 and
And keep it there.
there I go.
Want me to slow down?
Speeding up.
Okay, cool. Bob, for the mezzo is not the express, but the one below it.
Yeah, one below it.
Is it?
I think it's the
Yeah.
top one.
You
Yeah,
think
the
it's
one
top?
that's selected. I believe
Okay.
so.
Is yours on?
Yeah. Yep. We're
Okay.
all. Everybody's on. The whole gang's there.
Let's restart it. What's that?
You just hit run.
Huh? Oh, when run was the other one come up then?
Yeah, it'll
Oh, okay.
find it.
All right. Thank you.
Should find it. Then it'll automatically switch to 2.
Okay.
You can also click the run up here, but it's not as convenient.
No, I can't see it. It's
Right?
like, it was a whole game changer though when I realized I could do that down here with a mouse instead of like trying to fiddle with it up there.
Yeah.
Ah, there we go. Thank you.
Where's Herc going? That's the wrong direction.
All right, we settled in?
Early.
Hmm?
Yeah.
Feeling good?
Mm-hmm.
Sounds good. All right, back row, we're all settled in up here if you guys want to talk about the dive or anything like that.
Great. Okay. Well, good morning, good evening, good afternoon, depending on where you're watching from. We had a great dive yesterday at San Juan Seamount. I hope all of you got to peek in, especially at the end with our beautiful coral and sponge gardens. We have transited overnight to what we're calling Site 6 on Northeast Bank. This is an area that we expect to have ferromanganese crusts coating the rocks. And we, as in the last few days, we are out to characterize the biological and microbial communities associated with these substrates. We will be starting at the base of the escarpment or slope in what we hope will be sediments, and we'll be coring there. If they aren't sediments there, we have a waypoint further, I guess, to the east that should take us back into some corable habitat. And Paul Jensen's after microbes there with biopharmaceutical potential, and our lab probably will take some cores as well to characterize the infauna. There's also a student interested in microplastics in these cores. Then we will be traversing the slope, rather steep slope, we hope, covered with ferromanganese encrusted rocks, and we will be sampling the rocks to quantitatively analyze the smaller animals that live on the rocks, and we will be sampling the larger biology, these corals, sponges, bryozoans. Paul Jensen's lab will be, I should say, for looking at microbes with unusual potential that might aid in curing diseases or reducing symptoms, or even of industrial value. So we'll be taking samples of the animals, the rocks, we'll be sampling water to compare the microbes in the water to the microbes in the sediments and the rocks and the animals. And we'll be traversing hopefully all the way up to the rim of Northeast Bank. Want to say anything, Kira, about the rocks?
Thanks, Lisa. You covered all the exciting parts about the minerals. I read up a little bit about Northeast Bank this morning in terms of the formation geology, and there were 3 stages of volcanism in the Southern California borderland, which is the region that we're generally observing in this cruise. And Northeast Bank is actually part of the 3rd volcanic phase, and actually San Juan Seamount was formed from the volcanism at a similar time period. So even though we're further inshore and we'll see some different mineral environments and a little bit more productive biologically here inshore from San Juan, it's similar formation processes for these edifices. So it's going to be a great dive. Hopefully we see lots of nice thick crusts and some cool associated biology.
How old is the feature, Kara? Do you know? Or is it in the 7 to 10 million range? Or
Yes.
So if you guys have HYPACK survey visible, just kind of an overview of our dive. I know it's hard to see the waypoints, but they track, if you could see my mouse, kind of directly up through here. So we have an S point and then a T. We're actually launched on T. S is just an alternative. If we don't have enough sediment here to collect cores, we can back up to S. But then we want to kind of start heading upslope. The primary points in the dive plan are S, T, U, V, and we've added a few intermediate ones just to kind of help us have goals on our way up the slope. So if you see TA, TB, etc., they're just the points between T and U. We have slope, bathymetry, and backscatter available to look at.
Thank you, Aaron.
Yeah, it's tough being a dog and a scientist at the same time. Costume malfunction.
Noses and microphones.
For those at home who can't actually see the control van and the people in the control van, the back row has an anemone, a dog, and an octopus, and the front row has a devil. I can't see from the back very well, and a
Jaguar shark.
shark. Yeah.
Jaguar shark. Very, very particular.
So happy Halloween, everybody.
Happy Halloween.
Is a jaguar or shark a thing?
Nope, not at all. It's from Life Aquatic.
It is now.
It could be a leopard shark.
Nope, jaguar. 'Why'd you do that?
This time's going to seem so straightforward
make
compared
pictures.
to yesterday without all the deployments.
Hard no.
One 1319.
We've just passed through 400 meters and the oxygen's starting as declining, getting towards the edge of the oxygen minimum zone. There's a lot of migrating plankton that don't go much below 400 meters because of the low oxygen. And maybe the jellies were just hanging out at that boundary, eating things that were migrating down today. Some animals will migrate all the way into the oxygen minimum zone, but others don't. Another jelly.
I like that.
The other thing we've discovered is when we're on the slope that fish that live above about 450 meters will swim up and eat migrating plankton, but the bottom fish that live deeper feed on the bottom instead of swimming up, even the same species. So the oxygen seems to have some influence over the fish behavior and diet.
Like when you think about the migration and the water column, I just started just thinking about that because it was part of my thesis. I never thought about it before, but it is amazing for the size of the creatures that are going up and down, how far they're traveling every day. Just such an extensive use of energy.
It is. I forget. It was something equivalent to a person running 90 miles every day in terms of body length
Yeah.
and
Wow.
distance. So that amount of energy for us to run 90 miles every day, I don't think most of us could even contemplate that.
Like ultra marathon runners.
But every day, no
Yeah.
break.
Yeah.
That's how far those vertical migrators go. Translated to human like distances. Do they?
You're not on SPL?
Yeah, do they have a way to change their buoyancy?
Yeah, I think they do. I think they can sink at will,
Yeah,
but many of them
they
swim
have to
down.
swim.
Many of them swim down and up.
But they only, presumably they just have to go one direction. They don't have to,
Well, they go down in the morning and up.
but I mean, they have to extend energy to go in one direction and not in the other, maybe.
I'm not
Right?
sure. They may have to swim down and swim up. But they don't throw weights off when they're ready to
Yeah.
go up.
I don't know, they might poop. Are we allowed to say poop?
Yes.
I think so. Everybody poops.
Ah, that was one of my favorite books when my kids were little.
Oh, yeah.
Our whole family liked that book.
Yeah.
Shrimp. Argus.
So one of the things that's pretty interesting is that we're coming on to the same depth that we left San Juan Seamount from, and there's way less oxygen inshore here at Site 6 than there was at San Juan Seamount for a given depth. Maybe not way less, but definitely less. So we're at 1% saturation or below right now. 550 meters, and off much further offshore, I forget what the saturation was, 2 or 3%, something like that. Enough to make a difference. And it could affect why the communities will look different. We'll see.
Have you ever turned off the lights on the ROV to see what types of lights, bioluminescence, can you see in the water column?
Yeah, I think we probably go by too fast to really spot things. I don't think the cameras are sensitive enough.
We did, I think we messed around with the Brendan Phillips low light cameras trying
Yeah.
to capture bioluminescence.
But we can't just turn off our lights now and see anything.
Right.
Sometimes we turn off Argus lights to get rid of squid, because they like the light.
That's right, to get rid of the squid storm.
Yeah. It's like the birds, but with squids.
I think they're so cool. So many.
Yeah, we turned off Herc's lights to try and get them to go to Argus.
When Nautilus was in the med, we had turned off the lights on Hercules to see bioluminescence, and we saw some good light shows.
Really?
Yeah.
The camera's sensitive enough?
Yep. Even
Huh.
the SD cameras.
Wow. Huh. Want to see now?
Yeah.
Turn the gain up on it? Oh, you also have bucket cams off.
Are we getting a special Halloween treat?
Spookier the better.
Calling all bright shiny guys.
We've done it and not seen anything, too. I mean,
Yeah.
they're not always going to be there.
Yeah. All right. Lights on.
If we could kill all the lights in the control van, I will bioluminesce. I have glowy spots.
Huh.
She's not kidding.
Thank you. That was very fun to try.
I don't know if being on the bottom makes a difference, but when we did it, a couple of times, it was always on the bottom.
We got quite the current blowing up that way.
Wow, sure do. Big swing.
What was the verdict on the clogged slurp tube yesterday? Did we find the culprit?
Well, they got cup coral and a couple of things out of there. There was no sponge, though. We flushed it pretty good. Need to get one of those drain snake.
With a little camera?
Well, I don't know if we're going to get that fancy.
Colonoscopy for
It's hard to find clear hose that's sturdy enough for the slurp. It just, you can't really get clear hose that is heavy duty. So half of the hose is clear hose, and the part that sticks out the front is opaque.
So for all of our young engineers out there listening, we need some strong, clear plastic invented for ROV slurp hose.
It may be out there. I don't know where it is.
Go ahead, Bridge. What do we call the seamount we're diving on? Northeast bank. Yeah. You're welcome. God, don't you hate when you save something and you immediately can't find it? So frustrating.
Does everybody have a favorite Halloween movie?
It's a good question. I don't like scary movies at
It
all.
doesn't have to be scary.
I know. I'm trying to think of a non-scary one.
Like mine is Practical Magic.
I've never
Young
seen that.
Young Frankenstein.
Stein.
Yes.
That's a good one.
Yeah.
Oh, Shaun of the Dead. I like that a lot.
Oh yeah, that's good.
That's a pretty good one.
Back row, you got movies you like?
Maybe just the original Halloween.
Oh yeah.
I don't think I've ever watched a Halloween movie.
No, you never saw Hocus
Maybe
Pocus?
The Body Snatchers. That's dating myself.
I'm a fan of Beetlejuice.
Yes.
Oh, that's a good one.
Don't say it 2 more times, though.
Oh no.
Ooh, or the Great Pumpkin, Charlie Brown. That's also a classic.
Yes, it is.
It always made me so sad though when he never got anything. He just got the rock every time.
Well, I heard that that's not going to be on TV this year. It went to Disney. So you got to have Disney Plus to be able to watch it.
Not cool. That's like it's like an American tradition. Like you can't
Yeah.
just take that away.
Should be public access, yeah.
Yeah, I think this was what the first time in like 50, 60 years that it's not going to be
Yeah.
on public television now.
Wow. Hey Bob.
That's like making The Wizard of Oz of, Hulu.
Yeah.
We should have an amendment to the Constitution preventing this.
Rudolph the Red-Nosed Reindeer. Imagine that.
No Nightmare Before Christmas
you
fans?
can go to.
That's
Yeah.
another
Ooh,
good
another
one.
good one.
I've actually never seen that.
31, I'll slow down.
I've never
Let me
seen
go.
Nightmare Before Christmas.
Such a cool movie. Very, very cool visual.
One year we had a one-day student cruise on Halloween. We had a costume contest and 5 people came as anglerfish. We had a school of anglerfish.
What did they use for the light?
All sorts of little tubes and things that light up. Not everybody lit up, but they definitely had the little hanging thing in front of them. One person was a tinophore that glowed on and off with Christmas lights.
Ooh.
I like mix mashing costumes, so like a Tina 4, but like a Tina Turner-4.
Oh,
Yeah.
now you're coming up with all the good ones.
I know. I got coffee in me, so I'm starting to boot up.
We had a threesome that came as a methane seep, and one person was the tube worms, and one person was a Yeti crab, and the other person was covered in Taco Bell wrappers, and they were the methane seep. Taco Bell bean burrito wrappers. Yep.
All right, targets and
Look at that.
look at that. Let's make them purple. No, too dark. Lavender. Oh.
Are we coming down somewhere close to the launch target?
I'm gonna check now. Just how to get some targets in to the pilot software.
Oops.
We're a bit off. You can't get back though because of current, can you?
It's blowing me around. I can try and lateral some.
We're headed towards, is that right? Oh, T must be buried or I didn't add it. Gosh darn it.
I gotta speed up because now I'm.
So when you lateral your vertical,
Yeah, I'm speed up.
okay. I slowed down too, so it's give and take, Bob.
Yep.
That's about where it
Oh,
wants to be.
there's T's up there though.
That's TA, so
Oh.
it's ST. Those are just intermediate points to kind of give us a goal. I'll go edit the file, but that's about where you're headed. We still have 300 meters. I can ask the ship to step up a bit if you like. Okay.
You're the navigator.
But I like to make sure that everyone's happy. Are you happy?
Sure.
All right. Bridge nav. Can we get a 20-meter step bearing 219? Thank you.
A few more fun facts about Northeast Bank, where we're diving today. It's actually a pretty large island, about 90 square kilometers, 200 up to 200 meters above sea level for quite a bunch of time. So it used to be an island and has since subsided due to cooling of the oceanic crust beneath it to be this cool bank fixture we're going to dive on today.
Do we know if people ever lived on that island?
How long ago was that?
Yeah. Might have been.
How many million years ago was that?
Well, it formed between 7 and 11 million years ago. So it's probably a little bit pre Homo sapien when it was when it was subaerial.
In San Juan Seamount also was above
Yeah.
the water level for a
Yep.
while.
Yeah, a lot of the features this close to shore were once subaerial.
This might be our deepest dive so far of the series, I think. Now that we've been down to 1,300.
Yeah, 1,300, couple 100 meters below the other two.
Notice the oxygen levels are starting to go up.
Oh yeah.
Gone below 1,000.
So what makes the oxygen go down but then come back up the deeper we go?
Great question. The midwaters between a few hundred meters and 1,000 meters are the most isolated and the oldest waters. So they've had a lot of time for microbial respiration to use up the oxygen, and it's not readily replaced. The deeper waters have oxygenated currents flowing through maybe Antarctic bottom water. I'm not sure which the bottom water is here, but basically the surface currents don't penetrate into the oxygen minimum zone, so they don't bring oxygen. Things like the California current, which were even the undercurrent were California undercurrent were deeper, much deeper than that now. So these waters are just isolated. Some of this water hasn't been in contact with the surface in hundreds of, not 1,000 years. And then, but below there's influence of oxygenated currents. So even though it's inching up very, very slightly, we're still below. Well, what are we at? 1% saturation. We were at 1% saturation, and now it's going up. Those very small differences make a big difference to the animal life, and we tend to see really strong zonation across the bottom of the oxygen minimum zone. So I'm guessing as we head up slope from 1,300 to what our top depth is going to be, might be as shallow as 500 or 600.
503.
503?
If we get there.
Yeah.
Let's get there.
We got there. Yeah, we'll see a whole lot of zonation of animal life with different oxygen tolerances.
Oh, wow. Really cool.
Even 5 micromolar plus or minus can make a huge difference to the diversity and what can live there. However, the deeper we go, the less food there is.
Right.
So we may not see this profusion of life just because there's oxygen. And the other thing is once we get closer to the bottom, the oxygen gets used up a little bit more because there's more resuspended microbes and animals associated with the bottom using it. So I'm not sure. I don't think we'll see two minima, but see it has been climbing just a tiny bit.
Yeah. Almost to 1,200 meters.
It's picking it up, yeah, maybe. It's the same settings as yesterday.
Nice color choice.
There's like glitter in the bubble cam.
Like what?
Like it's like glitter in the bubble cam. You see it? Or maybe it's just things reflected off the lasers.
Yeah.
Never mind.
Have you ever seen the lasers stimulate the animals to luminesce back?
I don't know.
I don't know. I don't know if I can remember.
I was just thinking about the fact that this ship is full of people wearing masks, so we're all ready for Halloween, pre-adapted.
Yeah.
When I was trying to find this mask for my shark face, there was a lot of interesting options out there. Lindsay's clown mask is pretty creepy.
Yeah.
I like Lindsay's cape.
His seamount cape.
He's got a bathymetric cape with all the colors.
That is a bathymetric cape only to the likes of us.
What is it to everybody else?
A pride flag.
Oh, a pride flag, okay. I should know that.
Yeah, for the world out there, our mapping coordinator is dressed up as a seamount, wearing a rainbow cape because our default color map for our bathymetry is rainbow. And then there was a special seamount that he feels had a clown face on the top, so he's dressed up as a clown seamount.
I'm at 30 meters above the bottom.
Okay.
I'll stop.
Yep.
Almost there.
Auto heading on.
Go ahead, Bridge.
Wait until you get under.
No, you can do it now.
Okay.
Okay, you can just come down with me.
Yep. Coming down.
Almost there.
Ew.
Sea floor.
There she goes.
Ah, several fish.
Nice sediment.
I think that's plenty sedimenty, don't you think?
Plenty. Yes. No need for waypoint 1.
Cool. Check that one right off.
Is that okay?
Yeah, you
Cool.
can turn around and look at me.
Sounds good.
Tilt down or whatever.
I'm tilted down, so I'll just turn. Wrong direction. Oh, okay. It'll help if I turn my thruster on.
Let me know when you're good for reset.
Yeah, you can do it.
All right. Okay, reset. Good to go.
All right.
Yep.
Well, Paula's excited. A lot of mud.
Plenty of mud.
See if we take off here in auto exploring.
Hey, this is Caitlin from down in the lounge. Could we pop a Niskin before we land?
Copy that. We're at the seafloor. We're not going to touch the seafloor quite yet. Can you pop a Niskin before you put it on the seafloor? Thank you.
they're still adjusting and getting everything ready for the operation. So
Are we zoomed out?
Yeah, but we'll do it before we kick up some dust.
okay.
Zeus is zoomed out.
Yes, we are.
Yep, you are.
Hey, can you turn on the porch light?
Oops, sorry. I thought you were on to that. Yep. Light. Porch light's on.
All right, what sample number is this data?
This should be NA124-057.
Okay, thank you.
That's why. Hey, you watching?
Yep. Watching. You're
Popped
good.
it.
Fantastic. Thank you.
No problem.
Is the depth 1287 or 1297?
1297. It's 1280, but he's up in the water column.
Yep.
So I add on the
1290.
altitude.
Okay. Thank you.
Can I get a repeat of how far off the bottom that Niskin was collected, please?
9 meters.
Awesome. Thank you.
No problem.
Hey, out.
Could you remind me what this is for?
This is white balance. White
This-
balance for the camera.
Ah,
It's so the camera sees all the colors the way they actually are. Our eyes do it
It
automatically.
happens at the beginning of every dive and sometimes in the middle at the watch change. When we're all ready, we'd like to do a series of probably 8 push cores.
Okay. Any old place?
In the bottom.
Not mid-water? No mid-water?
No. Actually, would you mind if we did it in Fiji? I was supposed to be on sabbatical there.
How disappointing.
You might be doing that in the next couple of years.
You guys. Oh,
Yeah.
that'd be good. Okay, I can join you.
in on the Argus view? Kylie, can you switch over to sample mode?
Yeah. Can you do it? Sorry.
And then drawer out?
Tool tray out.
No, sample tray.
Sorry, yes, yes, yes.
Oh.
Too far.
No, I got it. I got to fix something here. This got fiddled with last night, and the gripper's not really closing. So,
I would have been so impressed if you guys opened the drawer and like a snake popped out, like a Halloween surprise.
Oh, why don't we think of that like little scary face in there?
At the view, there's Bob, and then all the hats peeking over.
Oh, nice. Yeah, I can see your heads up there.
Unfortunately, I could not function with my mask on the whole time.
I got to do this.
How can I help you?
I thought Bob was calling God,
Yeah,
asking for some help with
little
Well,
fun.
I am. I'm
manipulators.
I'm calling up an email from the guy who built the craft arm in order to set the grip force.
Phone a friend.
Oh, wow.
Select the grip force level you want to adjust. 1, 2, 9. Grip force. 9. And then what? Press setup. Then gain. Then 7645.
I don't know if that's helpful.
Oh, we're at 7% oxygen saturation down here.
Hi.
Yeah.
Interesting.
The seafloor is not covered with fauna the way it was on at Site 4. No sea pen. We're not seeing sea pens, sea pigs, turtle stars in large numbers.
It's going. It's a little bit closed now.
Well, that's not going to do
That's
it.
not that wasn't what you asked for. Oh, no.
Okay, wait. One more time. Grip, normal screen. Inner halt. Select the grip force level. 9. Let's try 8. Then set up gain.
All right, looks like we're grabbing
there
that,
it goes.
getting ready. Oh, testing the claw.
Claw.
It
The claw.
did close relatively quickly that last time.
We were watching you guys trying to calibrate it, I guess, last night from the data lab.
Yeah.
Yep.
And he had like a yellow tube in his hand, and we're like, oh no, not the umbilical. Like it looked like Herc was gonna make a run for it. He's kind of so free. Come on, Kra.
I don't know what's going on here.